Hello everyone, welcome back. My name is Arias, and today we're going to be talking about why I think Ginkgo Bioworks has a tremendous chance at being successful long term by talking about a few key tenets of the bullish thesis around them. Thanks to Simon Barnett of ARK Invest for writing on Ginkgo Bioworks, which I leaned on heavily in the making of this video. We'll also hear from one of Ginkgo's co-founders, Reshma Shetty, on a particularly interesting portion of Ginkgo's development process. This video is not meant to be a comprehensive deep dive on Ginkgo Bioworks, but rather a deeper look into what I think will make them successful. If you are looking for an introduction, I'll link mine below, and then come back and go a little deeper on their technology. Don't forget to like and subscribe if you enjoy this type of content, and leave any video suggestions and feedback in the comments below. And without further ado, let's get into it. The first element of Ginkgo's success is their horizontal platform approach. There are three broad steps in the synbio value chain, those being discover, engineering, and manufacture. Ginkgo has become the leading experts on the first two of these steps, but will also support customers who are transitioning into step 3. This is what having a horizontal platform means. Ginkgo is focused on being excellent at discovery and engineering for any application, rather than being focused on bringing any specific product to market. This both simplifies their focus internally to be solely on successful development of cell lines, rather than commercialization of these products, and drastically improves their efficiencies that can be derived from scale. Because Ginkgo is working on a whole bunch of different development projects, they are running a ton of experiments all the time, which lowers the cost of each experiment. Compare this to traditional companies that are attempting to use synthetic biology. They won't start out at scale and won't make the investment to build all the automation facilities that Ginkgo has, and will therefore have higher costs for each experiment, in addition to not having the same level of expertise that Ginkgo's engineers, or cell developers as they like to call them, have. This creates a clear selling point as to why customers should use Ginkgo's platform. Ginkgo's platform is less expensive to use and will only improve as Ginkgo brings on more programs and scales the number of experiments. Another key tenet of Ginkgo's success that we will see recurring throughout this video is that each of these programs and experiments contributes to Ginkgo's codebase. Improvements to the codebase improve the chances of success for each of the other programs in development and all future programs. This success will attract further customers. More customers means more throughput on Ginkgo's laboratories, which lowers costs, bringing more customers who will contribute to the codebase, and so on. This is a key flywheel that Ginkgo has started, and I think few understand it today, but it is key to the bull thesis around DNA stock. On this side, we will take a break from talking about Ginkgo's bull thesis and talk about one of the parts of the short report put out about Ginkgo late last year from Scorpion Capital. They took much of the report to focus on Ginkgo's platform ventures, which, simply put, are companies that Ginkgo has spun out and then use Ginkgo's platform to design a strain for a particular purpose and then be in charge of commercializing it. Scorpion hated this for many reasons, but most having to do with so-called round-tripping of revenue, where Ginkgo would invest in a platform venture, the venture would then spend the money with Ginkgo, showing revenue, and then Ginkgo would take that revenue and use it to invest in the next funding round, after raising money themselves based on the higher revenue shown. While this sounds bad, a 15-year-old company having to round-trip investments to earn revenue, this isn't how I see it. There are many reasons for the formation of platform ventures. One being that, earlier on, synbio was brand new, and the space had had no time to develop, and Ginkgo had not proved all of their capabilities. Being a horizontal platform, like I talked about earlier, they weren't going to develop their own cell lines in-house, so to find some of their original customers, they had to create them from within. As long as Ginkgo's platform works and the developments are successful, then these will be tremendously valuable investments for Ginkgo that will be great for long-term shareholder value. Another reason for the formation of the platform ventures is they allow outside capital to invest and grow the amount of capital in the synbio space without diluting Ginkgo. Platform ventures also allow for another avenue to acquire the most valuable and elusive asset of all, talent, who may be attracted to a specific problem, but who would not be interested in working at Ginkgo. As a final rebuttal to this gripe that really hit home with me from Simon's article, is that it would actually be much more frightening if Ginkgo wasn't willing to invest in companies that they partner with on development. If they weren't willing to invest, that would signal that they don't fancy their chances of success very much, which is clearly a bad sign for the actual usefulness of their platform, which is the true important factor long term. Ginkgo's eagerness to invest shows the confidence that they have in their platform to develop a solution and in synbio long term. However, as synbio progresses, I expect Ginkgo to have mix shift away from platform ventures and towards unrelated parties, as it becomes obvious what a deal it is to take advantage of their expertise and develop on their platform. You can start to see this take place as Ginkgo has signed many unrelated companies over the past few years in a variety of different sectors, as you can see here. Feel free to pause the video and look some of the most interesting ones up. Here lies another benefit for Ginkgo and their partners that comes from their choice of a horizontal platform. Ginkgo has the flexibility to develop for companies at all stages and tailor deals around the company. The main dichotomy is between young small companies who may opt to pay Ginkgo for development with equity, and large established companies who may want to pay more money up front for development with bigger milestones and smaller royalties. However, the deal structure, all of the learnings will still contribute to Codebase and contribute to Foundry scale, and this flexibility will only help Ginkgo get more programs on board faster. Moving back to Ginkgo's platform, but this time through the lens of one of the 5 co-founders and Ginkgo's CEO and board member, Reshma Shetty. The whole interview, which I've linked below, was excellent, and I'd recommend it for anyone who is interested in Ginkgo, but we are going to focus in on just one segment that I will play for you in a second, where Reshma explains a very interesting part of Ginkgo's platform. In the lead up to this segment, Reshma was talking about Ginkgo developing cells in a similar manner to how any company develops a product using the design, build, test, and learn cycle. Of course, this is much more difficult as biology is much less predictable than, say, physics or programming, and takes much longer to test as organisms take a certain amount of time to grow and exhibit the desired traits. There were 4 ways they talked about improving this cycle. You can improve the design process by looking to nature or to your codebase for functional pieces of code that may have the desired effect on the cell line. Another way to improve your search process is using computational methods. While biology is still unbelievably complex, computational analysis, especially some of the newer machine learning techniques, provides some help in biasing their search towards the desired outcome. The third way to improve the DBTL cycle is to parallelize your search and do thousands of tests on thousands of different designs all at the same time. This is where Foundry really comes in handy, as Ginkgo can do these tests for a significantly lower cost than competitors, which improves the chances of success. The final avenue for the improvement in the DBTL cycle is what I found most interesting, and is what Simon termed adaptive laboratory exploration, or ALE. Let's hear Reshma explain it.
Those are certainly 3 important families. I would say the other one that you didn't mention, but is also really useful, is leveraging evolution. So biology is pretty good at searching this space for functional designs. And so you can also, for example, generate diversity and then use certain evolutionary methods to let biology find the working design for your application of interest.
Oh, okay, that's interesting. How does that work in the context of Ginkgo?
So, for example, say I'm interested in a cell that can eat a particular nutrient of interest.
Mm-hmm.
And maybe this is not something that cells normally eat, but if you could get it to eat, like, hey, it would degrade this like waste product, or it would provide a new carbon source, a new feedstock for your application of interest. Well, what if I essentially generate a bunch of diversity? There's different ways to do that, but I'll just say I'll generate a bunch of diversity, a bunch of different cells with different designs in them, all of which are around a pathway for eating this feedstock of interest. And then what if I put those into a vessel and I give a limiting amount of that feedstock to the cell, so that the cells that grow the best, the cells that can consume that feedstock, extract energy, extract carbon, they're more likely to replicate and win, right? And so this isn't always available to every application, but there's sort of a lot of clever tricks that people have come up with over the years to sort of link up evolutionary pressure to a particular functionality of interest, and so they can use selection to get to a working design.
Essentially, just as you heard Reshma explain, ALE is the process of linking an organism's ability to survive and thrive to a desired trait. I believe this optimization is made by making thousands of different iterations of an organism that may achieve the desired output, but with small differences, then placing them in an environment together with selection pressure for the desired trait. Then, naturally, the strain who best exhibits the desired trait will rise to the top and dominate the colony. Simon Barnett also addressed ALE in the aforementioned article on Ginkgo. He advised that ALE often lends itself to minor iterative improvements each cycle, but can occasionally break out into very strong, fast improvement, as seen here. An example of where ALE was used was in the very successful development of Motif Foodworks protein production yeast strains. To wrap up the section on ALE and give another cool visualization from the article, Ginkgo's goal can be surmised by saying that they need to move the strain from point 1 to point 2. They need to do this as quickly and reliably as possible. However, this is a highly fraught endeavor. As Simon points out, there are many false peaks in the sequence space that may lead to the developers prematurely declaring victory, as any change will look like a step down with respect to the desired trait. All the while, the true peak could be hiding just a few tweaks away, with significant improvement in yield to gain. Furthermore, there are many absolute troughs, which further complicate development, as any changes will result in an increase. However, the cell developers must discern which direction they should make the tweaks that will move them as quickly as possible towards the peak of the desired trait. This is the challenge that cell developers of Ginkgo must master, and it is no small feat. Now imagine all the other teams trying to tackle this challenge without all of the learnings of Codebase, scale of Foundry, or years of experience from working on this problem. They really have their work cut out for them. For the next 2 slides, we'll go through some random advantages of Foundry and Codebase that I thought were interesting enough to highlight. First is that Ginkgo bought a genetic synthesis company, Gen9, back in 2017. This is important for many reasons, but one of the primary reasons is it lessens their dependence on outside suppliers like Twist Biosciences. It may shorten their turnaround time for experiments, which is crucial for their learning speed. These are the type of investments that only companies like Ginkgo can make because they can see the long-term payoff, whereas other non-SynBio platform companies may not use the feature enough to get a return. Another thing is that Ginkgo is so deep into the development of Foundry that they have come up with proprietary liquid handling techniques. Each of these developments, like this, will make it even harder for someone in the future to come along and improve on what Ginkgo has already built. On the right, you can see 2 graphs showing the increase in lab operations and strains tested increasing on a long scale. This test leads to cost declines, as you can see here on another log scale. One thing to take from this graph, besides the clear cost improvements, is that it took Ginkgo 2 years to get below the by hand cost. What is not shown is that it took them many more years, as the company was started in 2009, and the founders have been working together much earlier than that. Who else is going to make the investment that they have and develop a platform as efficient as theirs at this point, when you can just partner with them for a fraction of the cost? And not only do they have to match this level of spending efficiency, but it's a moving target, as Ginkgo is driving hard to continue these level improvements. I'm interested to see this graph updated going forward, as it is an important indicator to see if they can continue these cost declines. Now for some random advantages of Codebase that I thought were interesting enough to highlight from Simon's article. Some diminish the value of Codebase by saying there are plenty of open source databases that have similar information to what Ginkgo has developed. This is pretty clearly wrong in my opinion for a variety of reasons, one of those being that they use 2 different types of long-read sequencing, both Oxford Nanopore and PacBio, in their development, whereas the vast majority of open databases only use Illumina short-read sequencing. Using long-read sequencing allows them to better characterize repetitive microbial genomes. Another differentiating factor of Codebase is that it is made up of real data, meaning real tests and real results of those experiments. This is especially important because genes are extremely unpredictable. A sequence that works in one place for one function may not work the same way in another context. This is why it is imperative to build your own dataset. Ginkgo was only able to develop Codebase through the massive scale via automation that Foundry brings, and this will make it extremely difficult to copy. Furthermore, Codebase improves as more development occurs on the platform. As their Codebase improves, less complex developments will become easier and faster as they gain the know-how from each of the new programs on their platform. As I have said, Codebase knowledge also improves the chances of success for all other programs. In my opinion, this recursive building effect will be huge for Ginkgo. And as more basic developments become more routine, this will allow Ginkgo to continue to push the frontier forward in organism development. This also starts to solve more problems with having a vertical platform rather than a horizontal one. Imagine if ABC company, who makes a type of food coloring, decides they want to improve their manufacturing progress using some synbio and decides to do the research in-house. They'd have to stand up their own lab and hire a bunch of PhDs and get to work. Assuming that they are successful, which is no easy lift given the work, once completed, they may continue to optimize their food coloring production strain to get a better yield. However, it's unlikely that they go after any other different product types, like say CBG production or vaccine capping enzyme production, because they are a food coloring company and that's what they do. This means that all of their learnings that it took to get their strain where they wanted it will never be reused. This is super inefficient as opposed to outsourcing the work to Ginkgo, giving up a little of the upside, and using everything Ginkgo's learned from a decade plus of cell development, in addition to the scale improvements that come with Foundry. Codebase is, at its most basic level, a bunch of DNA sequences that Ginkgo has detailed performance data on for each of their cell lines. But in reality, it is the know-how to make cell development work, and this is what is really valuable in synthetic biology. Now for my conclusions. I think the flywheel that is created by scale is what's going to win in synbio. The scale of Foundry is going to allow Ginkgo to learn more, faster, all for cheaper than competitors. Foundry allows Ginkgo to keep moving forward at a breakneck pace and add all of their learnings to Codebase to improve the chances of success and fast-track the development of all future programs. All of this because they have bridged the chasm of making automation cheaper than the by-hand cost. This, in my opinion, will make them damn hard to catch, as other companies will struggle to justify making these investments when Ginkgo is so far ahead. Finally, with regard to their flexible deal structure, I love this part of the business and I think it has tremendous potential, especially with taking equity. I normally hate how every platform nowadays is instantly compared to AWS, one of the fastest growing, best scaling, and most profitable businesses in history, but I'm going to do it here anyways. What if AWS would have taken equity stakes in startups that build on their platform? They would have stakes in companies like Slack, Airbnb, Netflix, Uber, Spotify, and Stripe, just to name a few. Needless to say, that would be massively valuable. Or what if Intel had taken some equity for every company that they had developed chips for? Even a 3% stake in Apple, which would be worth more than 3% by now from all the buybacks, would be worth around $75 billion. That'd be a pretty significant portion of Intel's total roughly $190 billion market cap today. Not to mention all the dividends paid over the years. Ginkgo has developed a premier platform in synbio, and thus is going to have the opportunity to create massive upside. The magnitude of this upside is hard to quantify, in my opinion, because we don't know what they could possibly create. I know it is tough to hang on in such destructive markets, let alone buy more, but this is where fortunes are made. All of that being said, I'm still buying. Thanks for watching. What do you think of Ginkgo Bioworks? Are they destined for greatness or bankruptcy? Let me know in the comments below. All suggestions and feedback are greatly appreciated, as always, and I read all of your comments. Thanks again for watching, and have a great rest of your day.